Welcome back to the Code of Life, and today we are talking about genetic annotations. This is the last step in the process of finding potential treatments. You can remember we had started with the DNA sequencing where the sample is digitalized. We came to the alignment phase where all these small DNA reads were mapped to a reference. We already completed the variant calling, and now we have the list of variants, and now we need to work with that. So the last phase is up there where the doctor is the end and get some information how could look like a good treatment or what information gets into the treatment course based on the DNA of the patient's sample. First of all, what is the purpose of these genetic annotations? Genetic annotations need to assess the impact of genetic changes. And again, keep in mind, not every variant is really a mutation and has a functional impact. Therefore, you need to solve what is a variant which is maybe compensated by the redundancy of the DNA code and which are potential mutations having impact or potential impact on the cell products. So the input for that is, of course, you see it here again, the results of the last variant calling process. It's the list of variants, and this is the structured format of that. And typically, we have talked about the format, but you see here the ID in the middle. The idea here is RS1 whatever. This indicates there is already some known information on the internet about this certain kind of variant. Because there's a huge database, for instance, the DB-SNP database, which curates this information, and therefore you could easily look up this website and see how popular this variant is in the whole population or how many indications are associated with that. And this is exactly what you will do in this phase of the process. The challenge today, if you have quite a lot, and I only put some of the icons here, quite a lot of these publicly available annotation databases. They are typically curated by research communities and they are very specific for one indication or another. The challenge now for the oncologists or the researchers supporting them is to use all of these individual databases to find the relevant information. It's again a big data challenge because you have the data distributed in different sites. You have to visit all of them. You have to check, is there different information? Is the information accurate? Is the information leading to the same path or to a different path? And this is a time-consuming and manual task today. Due to the fragmentation, you will also have sometimes one side saying this and the other side saying the complementary stuff. So therefore, you need to understand the differences and evaluate them step by step. And keep in mind, we are talking about hundreds or thousands or even millions of annotations, and therefore, this is really a challenging task. So typically what is done today, doctors typically check for one certain gene, which is known as a so-called oncogene, which is associated to the oncology disease here. In our context, we have a list of oncology genes which are associated to the lung cancer case we introduced. And if you see some alterations there, you will check if these are functional, and if they are functional, you will also check if there is a kind of personalized treatment addressing exactly this one change in the DNA. If this is not present, you will not be able to handle this, and typically you will not have the time to focus on all of these found annotations. I'd like to give you now a kind of understanding how complex this task is and how time-consuming it might be. Let's start with this one single variant we identified in the phase of the variant calling. Here again, this is one of the sites, it's the so-called DB-SNP, which is provided by the NCBI. And you see there's an easy URL where you can look up this stuff and you get to a page, and this is only excerpt, HTML website, and you see some information like on the top right, you see something like with pathogenic allele. That is important because someone already proved, or at least in the database, someone says that it's already proved that there is a change which might have a pathogenic impact on the body. This is important for the treatment because it's known to have a real problem or results in a real problem in the product of these cells. Again, a little bit deeper, you see something like this information here. This gives you the understanding. On the left-hand side, you see the kind of mutation, for instance. We see the T2A change. This is exactly what we also found in our variant calling. And this T2A change results in a functional change, and this is shown on the right-hand side, the change in the amino acids, right? And you see something like V is changed to E, and the position is the 600 position in amino acids. So it's a called V600E change. And down here, and this is another excerpt from the webpage, you see something like the how often this kind of mutation was detected in a larger community, in a larger population. You see it here. Here's a sample count of about 120,000 samples, and you see the A was only present in less than 1%. Most probably there's a T in this population. That means these changes are documented, these changes are rare. However, if they are present in the patient, you see they have pathogenic impact, and you see already that some kind of the amino acid product is changed. So what does it mean? The next step would be to understand how is this kind of gene involved. We are talking here about the gene BRAF. This is the location where we say this is associated, for instance, with certain cancer diseases like lung cancer, but also breast cancer and other ones. You see here, this is a complex map, and I don't want to go too much detail, but this is a so-called pathway. A cellular pathway which is curated, for instance, by the Keck Institute in Japan. They provide information, curated information, medical knowledge about certain genes and how they are involved in a kind of process. That means if there's a certain gene present at this site, it will initiate this and this kind of process. These maps are created by medical experts, and they will give you some understanding if the gene, for instance, BRAF, is changed or altered, what could be also affected in terms of processes in cells or products. This information is very valuable to understand the impact of a change. Another source of information, for instance here the gene cards, gives you a glimpse of an understanding what is the gene, the BRAF gene is involved in. So you see some meta details here on the top, where it is located, how long it is, what are certain oncogenes and so on. But you also get more information if you dig deeper, like these kind of genes might be also relevant for this kind of diseases. For instance, colorectal cancer, lymphoma, and so on. And this information is unstructured. This is text. You have to read it. And this is time consuming. Of course, most of these informations are publicly available and also present at doctor sites. But however, you have to investigate each of the gene functions, see how they are involved in the whole process. And then the next challenge is how to find a possible treatment. And typically you want to see the best possible treatment, the latest one which is published. For instance, here we took one of the publications at PubMed, and what you have to look now up is maybe there is the BRAF gene mentioned. For instance, BRAF hotspot mutations here in the title. You have to read the entire 6, 8, 10, 12 pages publication. You have to understand it, and then you have to assess if this is relevant for your patient or not. You see, it's really time consuming. And again, you need some tools to assist people, especially in a clinical environment, to make use of these information. And the second thing is, if you have these variants, most of these variants are not treatable by certain kinds of medications. But if you have a limited set of treatments available, you should be also linked to this medicine, this kind of therapy is targeting exactly this change. You need to start with this. This information can come from publications, but also from clinical trials. And this is really challenging because the amount of information which is created per day, some 3-4 publications per minute, this cannot be read by one single doctor at a time. So you need some processes which supports them in their daily life. So what's next? Next time we want to investigate how to guarantee the reproducibility of the findings, to establish a process which not only results into a list of variants, but you can do this every time in the same way and you come to the same results. That there's no one needed to do some taking data of one Excel spreadsheet, put it into another. And also how to enable a scalability platform for high throughput sequencing. That means you want to investigate not only one patient at a time, you want to investigate thousands of patients in your hospital or in your district at the same time. So therefore stay tuned, and I'm looking forward to see you next time at OpenHPI.